Okay, everybody, we're going to be jumping into the next part, membrane potential. If you didn't watch the first video about membrane physiology, you can go ahead and go back and watch that, or you can just go ahead and start here. I'm talking about more about the membrane potential. So when we talk about membrane potential, specifically we're going to be talking about something that's called an electrochemical gradient and the voltage that we get from that. Okay, so an electrochemical gradient, essentially what it is that there's a difference in charges inside of a cell compared to outside of the cell. Inside of the cell, we'll get a little bit more into this further down, the inside of the cell overall is going to be more negative and the outside is going to be more positive. And kind of that difference due to the different components that make up and that are floating around in the inside versus the outside of the cell are what gives it that charge. So different concentrations of ions, of cations and anions, as well as stuff like proteins and any of the other stuff that we have inside of our cells, make that difference, okay, and cause us to have what we'll call a membrane potential, meaning that there's potential energy because there's a difference in voltage. So here's kind of a graph of it, and different types of cells are going to have what we call a resting membrane potential, okay? That's just showing and saying exactly like what the difference is, like how negative those different cells are. So there are different levels of negative for like neural cells versus heart cell versus cardiac cells, the heart cells, versus muscle cells, skeletal muscle cells, and so on. And we'll look at a couple of those differences, just the ones that are kind of the most important that we deal with physiology. Big one to remember, though, is the nerve cells. Right now, here we have this set around negative 65 millivolts. Nerve cells sit right around there between negative 65 and negative 70 millivolts. So if you see that, that's going to be our nervous cells, or nerves, our neurons. Okay? And this graph here also shows something that's called depolarization and hyperpolarization. With all these cells, we have a resting membrane potential. So this is kind of where, when nothing is happening, what voltage those cells sit at. And that's considered the resting membrane potential. If it starts to go more towards more positive or less negative, we call that being depolarizing. This cell is depolarizing. And if it goes more negative, that's called hyperpolarizing. And we'll get into exactly why that's important, again, in a little bit when we look at something called action potentials within these cells. So the contributing factors to membrane potential, we have cell permeability and ion concentration affect membrane potential. We have the different unique ions, so for example, sodium and chloride, as well as potassium. So see, we have potassium, we have sodium, chloride, and also this A- right here is the amino acids, the proteins. Proteins tend to be highly negative, which is actually why inside of the cells we tend to have a very high negative or very low number, a very negative number inside of the cells due to that high concentration of proteins. Okay? So we can see here inside of the cell tends to be negative, and depending on what your specific professor has said, obviously go with what they say. But in all reality, the reason why the inside of the cell is so negative is because of the high protein concentration, and those proteins tend to be negative. All right. Another reason also we talked about here, we're looking at permeability to potassium and to sodium. So earlier when we talked about like those leaky channels and also that sodium potassium pump, those sort of things, that's kind of what we're getting into here. Specifically the leaky channels with this, where we have sodium and potassium kind of leaking back and forth. And we look at, so the permeability to potassium and to sodium, it is 25 times more permeable to potassium than it is to sodium. And we can look at that, we think, why is that? Okay? So when we think about concentrations and diffusion, we want to go from a high concentration to a low concentration naturally. So that's this chemical force for potassium. We have the potassium wants to leave the cell because it wants to go from that high concentration to a low concentration. There's also something called the electrical force for potassium. Because the inside of the cell is way more negative than the outside of the cell, that electrical force is kind of pushing back on the potassium, telling it to stay inside. Because we already have way too much positive here on the outside, it's more negative in there. And so the potassium is going to be attracted more towards the negative because it's a positive cation. So those forces kind of go against each other. You still get in general get leakage of the potassium out. Then for sodium really wants to come in, both because of the electrical force as well as the chemical force. So you do have a slow leak of sodium coming into the cells, and that's why we need that sodium-potassium pump. Remember the other name for it, it's the sodium-potassium pump, or the sodium-potassium ATPase. If we think back, what kind of transport is that? It's active transport. Is it facilitated or non-facilitated? It is facilitated. Facilitated because it requires a protein in order to work, and it is active because it requires the use of ATP. Okay? So, continuing, a lot of this is essentially saying the exact same thing. We have potassium sneaking out slowly and sodium coming in, because overall we have a negative intracellular fluid, ICF intracellular fluid. Again, this is just kind of repeating how the inside of the cell continues to become negative because of the potassium going out, and also because of the proteins that are already inside of the cell. So both of those factors are keeping it negative and wanting to have, and that sodium is then wanting to enter. And so we have a greater loss of potassium from the inside of the cell going to the outside than we do have sodium entering into the cell. So we have something that's called a steady state that develops. This is kind of like what you could almost call like an equilibrium kind of point. Where we get a steady rate, a very specific rate of sodium and potassium flowing, of this sodium flowing in and the potassium flowing out, along with the activation and the use of that sodium-potassium ATPase. And that is all of that together is what helps with resting membrane potential, what maintains us at that resting membrane potential. So specifically for neurons, the neuronal resting membrane potential, that's what this is down here, RMP stands for resting membrane potential. Remember, think back, what is that resting membrane potential? It's just that chemical charge that you have inside of the cell, kind of when you compare it to the outside of the cell. And it's negative because of all of the negative stuff that we have on the inside of the cell. So for the neurons, it's about negative 70 millivolts. This is something that you'll really want to keep track of and remember, and write down as we learn more of these specifically. You'll want to remember these to keep these straight, because sometimes we'll get graphs and different charts. And while a lot of this, the depolarizing and hyperpolarizing is similar between these, if you're given a graph and it'll show you exactly how negative the resting membrane potential is, that will help you determine what kind of cell you're actually looking at. Okay, so membrane potentials, there are two factors that determine membrane potential: the unequal distribution of the ion, as well as the membrane permeability. Membrane potential for a single ion. So this is an equation called the Nernst equation. Let me actually pull up something on my other PowerPoint, because I feel like it's described a little bit more on that other PowerPoint. Just give me a moment to find that. Okay. So we'll look at both of these. So when we talk about the Nernst equation, right, we also talk about kind of the Nernst potential, okay, or the equilibrium potential. It is the theoretical intracellular electrical potential that would be equal in magnitude, so in size, but opposite in direction to the concentration force. The Nernst equation is as follows. We have our millivolts equals plus or minus the 61 log of the ions in versus the ions out, as we can see over here. He has it written a little bit differently, but it's still the same exact equation. And so this is essentially what this does, is it tells us, based on how much concentration of the ion is leaking out versus how much concentration is coming in, what that potential is for that one specific ion. And this is just for potassium. If we wanted to calculate the cell as a whole, we'd have to do a bunch of the different Nernst equations for every single one of the different ions and the different things that are coming in and out of the cell, and add all those up, and that would give us kind of that membrane potential. That's how we solve for that. So, and that's actually why we have this other equation called the Goldman-Hodgkin-Cots equation, which considers multiple ions as well as the membrane permeability. So this is just looking at one single ion, and this is looking at the multiple ions as well as the membrane permeability. That's really all you need to know about these. Unless your professor says something otherwise, saying that you need to do specific equations and actually do the math, this is more just conceptual, understanding what these equations are used for. Okay, so the Nernst equation is used for membrane potential for a single ion. The Goldman equation considers multiple ions and as well as membrane permeability. So if you want to see like the whole big picture, you're going to want to use this equation. Again, don't necessarily have to have this memorized or anything, because in all reality, we're not going to be using this kind of equation for anything that we would need. And this is, again, like the actual equations themselves aren't really tested on boards either. Okay, so membrane potential contributing factors. We have that sodium potassium ATPase that we mentioned earlier, is what's really helping to maintain that RMP. Please remember this, that we have the sodium getting pumped out of the cell and 2 potassium brought in, and we are using ATP. So that means this is an active transport. And so the net charge that we have is a net -1 charge inside the cell, because we're pushing 3 positives out and we're only bringing 2 positives in. So it continues to keep that cell negative. And we think about that, that's really what we want, because if we have some potassium leaking out and sodium coming in through those leaky channels and just through normal diffusion, then we're going to be losing, because we're losing a bunch of we're gaining and having everything come in and out. We want to make sure that we maintain that level that we need to, that -70, so that way we don't get any accidental opening of those things that we looked about earlier called voltage-gated ion channels. Maintaining a good resting membrane potential keeps all of our different ion channels closed until they need to be opened, and they're open to the proper process. So the sodium potassium ATPase maintains the membrane potential as well as the cell volume. And just kind of the repeating all through this. This figure goes over exactly how that actually happens. I would take a moment and read through this little chart, just so you try and understand exactly what is happening step by step. It's good to understand that the sodium attaches first, and then once it attaches, it closes, and the ATPase gets phosphorylated with the energy. Then it opens to the outside, the sodium goes out, the potassium comes in, and then that binding of the potassium causes conformational change. Remember what that is from biochemistry. Changes in shape to the protein, and this is an intramembranous protein. And that change in shape is it then coming down and opening and allowing the potassium back into the cell. So gated channels control membrane permeability. So the leaky channels are always open. So that's going to be, because those are always open and there's no energy involved, this is going to be our passive facilitated diffusion, right? And then the gated channels open on demand, depending on the situation. We have mechanical, chemical, and voltage. A lot of what we'll talk about is chemical and voltage. We'll throw in a little bit of mechanical into there, but most of what we talk about here, especially here in the beginning, are the chemical channels and the voltage-gated channels. Okay? So when the gated channels are activated, open increases conductance. Let me pull up my other PowerPoint. When I took the course, we didn't really talk a lot about Ohm's law or anything. We did talk about current, though. So when these channels open, you can kind of think of it like almost like if you understand the way that a light switch works, when you flip a light switch, it connects the different ends of the wire to allow electricity to flow through, which then allows like the outlet or the light or whatever it is that you're turning on to receive the electricity and to be powered. So when these channels open, then you get conductance. Conductance means that like there's going to be flow of, in this case, of the ions, and that generates what's current. The flow of the charged ions, just like how electricity is flow of electrons. That is going to be your current. You have V for your voltage difference, and then R is resistance. And you have different kinds of resistance, where the plasma membrane resistance, the cytoplasmic resistance, and the external cellular fluid resistance. These are all things that resist the movement, kind of prevent the movement of those ions. And so they have to overcome these resistances in order to flow through. And this is just the equation. So current equals the voltage difference. That's the difference of the charges outside of the cell to inside of the cell, divided by the resistance, being all those things that resist the movement of those ions. And that will give us the current, the rate at which they are flowing through those channels. Okay, good 20 minutes. We'll go ahead and stop here with this one, and we'll continue on with action potentials.